Okay, can somebody confirm you can hear me?
I can.
Thank you. All right, today is May 13th, and we are supposed to be talking about Lab 8, identification of gram-positive organisms. I will send out your first aid on your unknown project sometime tonight, probably a couple hours after the lab ends. So it might be actually early tomorrow morning, but I might get it before midnight. But sometime around midnight, I'll probably get it out, maybe as early as 10 o'clock. Um, and let me remind you, your infectious disease paper is due this Saturday. Any questions about any of that? All right. I wonder if I have the lecture finished. I don't know. Oop, too quick. All right, Lab 8, identification of gram-positive organisms. We'll be talking about gram-positive cocci, especially Staphylococcus, Enterococcus, and Streptococcus today, as well as the gram-positive bacilli, Listeria, Clostridium, Corynebacterium, and Bacillus. There are some video clips in this lab, and in this lab, we're also going to look at a dichotomous key and case studies. The last two are provided in separate files in Canvas. Let me see if I can show you that. Ooh, looks like I shut that down by mistake. Come on, open up. I meant to shut down the tab and I shut down the entire. Boy, this is really slow. I must not have finished the lecture page, meaning the lecture must still be, the file must still be being made on my computer. Let's see if this opens. It does, fortunately. All right. So in the link to Lab 6, we got the lab module and then the worksheet for the lab. We also have the case studies, 1, 2, 3, 4, 5, 6, 7, 8. And then the dichotomous key. Um, I was giving these by the microbiology lead instructor, and I thought about putting them all together in one file, but I'm glad I didn't because he's changed one of the files. I think it was case study 4 was changed, maybe case study 5 also. Um, and I've decided to keep them as independent files so that way if they're ever changed, I can just put up the new file. Any questions about any of that? All right, let's go back to the lab module. The learning objectives on this lab is 1, understand the principles and how to interpret the results of the CAMP test, the Bialasculin Auger slant test, the SF broth test, and the Optogen sensitivity test. And then 2, be able to use a dichotomous key to solve case studies involving gram-positive bacterial infections. Let me warn you that the dichotomous key, which I just showed to you here, can only be used in this lab. Don't use this dichotomous key for your unknown project, do not use it for any other lab. It's designed specifically for this lab and only use it on this lab.
I don't think you're sharing your screen.
Oh yeah. Are
Thank
you
you for
sharing?
letting me know. Yes, I'm showing you things and you're not able to see it. You should have told me earlier. Right. Whenever I'm talking about the syllabus, I mean to share the screen. So if you're not seeing the screen and I'm talking about the schedule, you can remind me to show you there. So I tried to show you that the files to week 6, lab 6, the lab module here, the worksheet there, and then the case studies underneath it with the dichotomous key underneath it. So use this dichotomous key on this lab and this lab only. All right, when specimens are received in a microbiology lab for routine bacterial cultures, they are inoculated on a culture medium that will facilitate the growth of common pathogens placed in physical environments to support their growth. Microbiologists use colony morphologies and the Gram stain to provide important information on the primary classification of bacteria isolates. However, further biochemical testing is required in order to identify the species. In this lab, the CAMP test, the Biosculin test, the SF broth test, the optogen sensitivity test will be discussed. The CAMP test is an acronym of the three microbiologist names who invented or discovered this test. C for Christie, A for Atkins, and then MP from Munch Peterson. And they describe the amplified hemolytic effect that some beta hemolytic organisms have when they're physically grown near Staphylococcus aureus colonies. What you do is you take a single streak of beta hemolytic Staphylococcus aureus, let me blow this up a little, and streak it across a blood agar plate. So the white area there is the beta hemolysis caused by Staphylococcus aureus. You then take your test organism and you streak it in a perpendicular line to the Staphylococcus aureus. And then you look at which these will also be beta hemolytic organisms, which I really have to blow that up to show that with Streptococcus pyogenes. There you can see the streak, the kind of orangish darker thing in the center of this line, and then the beta hemolysis is around the Streptococcus pyogenes. The same with Staphylococcus aureus, it just grows bigger. Streptococcus pyogenes has no enhanced hemolysis when grown near Staphylococcus aureus, so it is just a streak of beta hemolysis. Listeria has partial or slightly positive CAMP reaction where we have slightly enhanced hemolysis, meaning a little arrow here. And there's the regular hemolysis, beta hemolysis. There's the Staphylococcus aureus showing you the beta hemolysis of that. So we have enhanced hemolysis slight for Listeria. And then Streptococcus galactea is positive in the CAMP test because it has greatly enhanced hemolysis near Staphylococcus aureus. And there's the Streptococcus galactea shown here in that, I don't know, orangish line, greenish orangish line. And then the hemolysis there is the normal beta hemolysis. And here we have the enhanced hemolysis that takes the shape of an arrow. Any question on the CAMP test? All right. The Biosculin hydrolysis test is used to differentiate Enterococcus from Streptococcus, those two different families. If an organism hydrolyzes esculin in the esculin agar, it'll make beta D glucose and esculatin. Esculatin has a dark brown color, and if more than 50% or 50% of the tube is dark brown, it's positive in the esculin um, hydrolysis test. If, on the other hand, the tube is negative, it'll have no darkening or very slight darkening of the tube, and that's negative. Streptococcus species are negative in the bile-esculin hydrolysis test, and the Enterococcus family is positive in the bile-esculin test. Any question there? All right. The SF broth test is named for Streptococcus faecalis, which is the old name of Enterococcus faecalis. They thought it was a member of Streptococcus family, but when they sequenced the bacteria, they discovered it was its own family, Enterococcus. But the SF broth test still is named for the old name of Enterococcus faecalis. Any question about any of that? And I'm just going to call it the SF broth test because the correct name is not Streptococcus faecalis anymore. It's Enterococcus faecalis. This test was used to separate out Enterococcus faecalis from other Streptococcus species. If the organism can ferment glucose and produce acid products, it'll change from the purple color of the broth to a yellowish color. So this is FF broth positive, and that is Enterococcus faecalis is SF positive. Other members of the Streptococcus family, in fact, all members of the Streptococcus family, are SF broth negative, and they will remain purple in color when the Streptococcus species is grown in the SF broth. Any question about that test? All right, the Optogen sensitivity test is a test looking at the bacteria's sensitivity to the antibiotic Optogen. This test is used to differentiate Streptococcus pneumoniae from other alpha hemolytic streptococci. So you have another member of the Streptococcus family. If it is alpha hemolytic, we can then use the Optogen sensitivity test to separate out Streptococcus pneumoniae from other alpha hemolytic streptococci. So what you do is you have the alpha hemolytic bacteria that you know is streptococci, which isn't shown here, but it's stated that the bacteria forms streptococci. It grows on blood agar. Let me blow this up even further. And there's the colony, kind of off-white. And you see this darkened ring around the colonies? That is alpha hemolysis. We then take this bacteria and plate it on an agar plate. After about 5 minutes, you then add the disc containing Optogen, and then you incubate the culture. And if it's Optogen resistant, alpha hemolytic streptococci, then it is not Streptococcus pneumoniae. If it is sensitive to Optogen, meaning it has a diameter greater than or equal to 14 millimeters, the zone of inhibition, then it is Streptococcus pneumoniae because it's the only Streptococcus which is sensitive to the antibiotic Optogen. I should say it's the only alpha hemolytic Streptococcus that is sensitive to Optogen. Any question about any of that? All right. Microbiologists use dichotomous keys that describe which tests should be or could be performed. It's using a logic where you have all possibilities, all possible gram stain, and then you split it into two choices, gram positive and gram negative. Let's say we're going to follow the gram positive, so we can scratch out the gram negative. And then we look at the shape of the cell. We have a gram-positive bacillus, a gram-positive catalase. We're going to cross out the gram-positive bacillus and follow the gram-positive cocci. We then look at the catalase test, which separates cells into catalase positive and catalase negative. We're going to scratch out the catalase negative and then follow the catalase positive, and this narrows it down to the Staphylococcus species, or the family. And then we can look at the coagulase test and look at the coagulase positive members versus the coagulase negative members. The coagulase positive members is only one, Staphylococcus aureus. And so if it's coagulase positive, Staphylococcus, catalase positive, gram-positive cocci, and gram-positive, then we know it is Staphylococcus aureus. Any question about the dichotomous logic and using a dichotomous key? All right, let's go to the lab exercises. You're not going to be performing any of these activities because everything we do will be done online. However, you will be held responsible for knowing how to perform and interpret the tests that we've discussed here. So view the following video clips, one discussing the SF broth test and the bile esculin test, and a third or second video showing you the CAMP test. And then let's go to the worksheet. Only fill in the worksheet and only submit the worksheet. Then fill in interpret the following tests by completing the chart. I've done the first row of the chart for you. You can get the information from this lab, but you will need to refer to some previous labs to get the information. Like a Schaefer-Fulton stain, stains for endospores. Tell me what the positive result for the Schaefer-Fulton stain is and what the negative result is. On this one, you have to go to a previous lab to get that information. And then tell me what the negative stain positive result is for the capsule test and the negative result for the capsule test. Tell me what the positive result is for the catalase test when you're adding the reagent hydrogen peroxide, and then the negative result. If you can, describe it in a word, but you could put it in a sentence. You can just blow that cell up if you need to. Tell me what the positive result is for the coagulase test and the negative result. Tell me what the positive result is for the oxidase test and the negative result. In this lab, we have the CAMP test. Tell me what the positive result is. And you can say enhanced beta hemolysis or just enhanced hemolysis. Just remember that all the organisms used in the CAMP test show beta hemolysis. So it has to be enhanced beta hemolysis. And the negative result, no enhanced hemolysis. Tell me what the positive result is for the bile esculin test and the negative result. Give me the positive result for the SF broth test and the negative result. Give me the positive result for the optogen sensitivity test and the negative result. Give me the positive result for the basotracin sensitivity test. And remember with this one, you do need to state a size for both the positive result and the negative result, meaning the size of the zone of inhibition for positive has to be greater than a certain size or less than a certain size for the negative result. For 3A, you don't need to do anything, but this is a video to explain and help you understand a dichotomous key. If you're perfectly comfortable with a dichotomous key, you don't have to do 3A. But if you're not comfortable with the dichotomous key, you should do 3A to help you gain familiarity with using and developing a dichotomous key. In this class, you're not going to need to destruct a key for this class, but it will help you understand the general behind the idea of using a key and help you with your unknown project. And then 3B, use the dichotomous key given in the lab. I've already showed it to you. And there's 2 pages in the dichotomous key. Let me show you that. There's the first page talking about the gram-positive cells. And you split it down to catalase negative. Let's go to the second page. There's the gram stain. This is for the gram-positive bacilli. Sorry, the first one is for the gram-positive cocci. And this is going through the gram-positive bacilli. We're going to be looking at these species or families here. So these are the possibilities which are in the dichotomous key. And then use the dichotomous key to solve the case study 1, case study 2, case study 3, case study 4, 5, 6, 7, 8. For the case studies, remember, they're here. No, actually, I got a better way to do this. Let me use from my lab because it'll be easier to, it'll open up better. All right, case study 1. A 13-year-old teenager attended a week-long summer scout camp. Upon returning home, his mother notices a pustule beginning to develop on his arm. Looks like a spider bite. The area is warm to the touch and becoming extremely painful. When her son starts running a fever, she takes him to a walk-in medical clinic for evaluation. The physician orders a gram stain of the exudate and a routine culture. Here is the gram stain. Let me blow this up a little. You can see the bacteria cells here, and the pink cells are actually the boy's cells. So tell me what the shape of the bacteria is and the gram stain. You can even give me the growth arrangement there. And this is the bacteria growing on a blood agar plate. There's the bacteria colony. You can see hemolysis on the blood. Tell me what type of hemolysis that is. In the lab, not in here. Don't mention it in class. Let me shrink this down now. I can get that to work. There we go. So those are the first tests. Then we have some more tests. They're taking the bacteria here, putting it on the slide, adding hydrogen peroxide. So that's the catalase test. There's the result of the catalase test. And then we're adding bacteria here on the slide, adding the blood plasma, and then waiting 15 seconds, and you can see the result of the coagulase test here. And then we have another test where we took the bacteria, grew it on the mannitol salt agar. It grew and changed the color of the agar. You can look that up. The mannitol salt agar, there's a test. Under the tests that you're using for your unknown. It's in one of the labs too, but we need to add this for this file. For some reason, your term doesn't have this added. I'll add the mannitol salt agar file here. Anyways, that tells you some information, and that's the last test, I think. And then what bacteria was isolated? You do need to know the species by going through the dichotomous key, and then write that information for case study 1. In case study 2, note the test 2, it just says test 2. It's the basic trace and sensitivity test. And the same for case study 3. So let me look at case study 2 just briefly. I'm trying to have a better way to get that. I can read the information. There's the bacteria, there's the gram stain. To see that, you need to blow up the slide to see what shape they are. And there's the catalase test. There's the results of the catalase test. This is test 2, the basic trace and sensitivity test. Is that right? Yep. And then test 3 is looking at the CAMP test and what bacteria was isolated. Any questions about the case studies? If not, go ahead and get started. I will be here until 8 o'clock to help you with the lab or until the last student logs out. So if you have a question, please ask it before the last student logs out to make sure you can get your question answered. Any questions? All right, get started on the lab.